This week's lab covers the enteric bacteria. So what are enteric bacteria and where do they grow? Well, the enteric bacteria inhabit or infect the intestinal tract. There's approximately 500 different species of enterics, and many of them are gram-negative bacilli. And in fact, bacteria make up approximately 60% of fecal mass. They're also part of our normal flora. What do? They inhibit the growth of pathogenic species. So if you've got all your good flora in your gut, in your GI tract, that inhibits, it doesn't give an opportunity for a pathogen to actually get a foothold and multiply. They also produce vitamins and they train our immune system. When they're pathogenic, they cause gastroenteritis. So in this lab, we're going to look at several things. We're going to use another selective differential media. We're going to use something called EMB agar, eosin methylene blue agar. It's selective, again, selects for gram-negative bacteria. The dyes in EMB agar inhibit the growth of many gram-positives, so therefore it selects for gram-negative bacteria. It's differential in that it helps us distinguish between bacteria that ferment lactose and those that do not. So this EMB agar has lactose in it. The bacteria that can ferment lactose are divided actually into two groups. There are those that are low acid production in fermentation, and we'll get bright pink colonies growing on this EMB agar. And then there are those that have produce a lot of acid in lactose fermentation. And then we get these dark black-blue colonies with a really bright metallic green sheen. And then other of the enterics, the gram-negatives, do not ferment lactose. And they're just kind of colorless or pale. They kind of have the background color of the media. So here's some examples. This is EMB agar. Up on the right, you can see that lactose fermenter, E. coli, it's a high acid production. We get this really shiny metallic green colonies growing. The top left is a lactose fermenter, but low acid producing. So we get this quite bright pink colony growing on our EMB agar. And then on the lower left is the non-fermenter. It's just sort of a pale, colorless colony growing there. So that's how EMB is selective and differential. Selects for gram-negatives and differentiates based on lactose fermentation. In this lab exercise, we're going to do a little bit of a mini unknown. There are 6 unknown bacteria. You're going to take one of them. We will set up these 4 different biochemical tests, and based on the results of those tests, on our next lab period, you should be able to identify which unknown bacteria you had. So we'll inoculate those tubes. We'll also inoculate an EMB plate. And just as we've done previously, you'll divide that plate in half. Half of it you will use for the unknown. The other half you will inoculate with your pet because we want to see how your pet behaves on EMB agar. And then when we come back on Wednesday, you will look at the results of these tests. And for my remote students, of course, I will be taking photos of those results and sending them to you.